Dear students, now we are into another topic in genetics, that is genomes to proteomes, genomes and proteomics. So, initially, some basics will see genes, a segment of DNA that contains instructions to make a specific protein molecule. So, the basic biological unit of heredity it is, and genes determine the inherited characters. We all know that. So this is one kind of an image which will DNA, the molecule of life, you see here. So genes in eukaryotes, genes are contained within the cell nucleus. The mitochondria also contain small subsets of genes. In prokaryotes, if you see, genes are contained in a single chromosome that is free floating in the cell cytoplasm. So these are the genes which are present in eukaryotes and in the prokaryotes. So if you see here, genes composed of exons, introns, and different control elements. Exon, which is a protein coding sequence, intron, it is intervening sequence. This image shows you that coding. So, if you see, this is also one image here which shows the eukaryotic protein coding genes. If you see the upstream region here, if you see, and the promoter region with transcription sites, translation sites, as I told you all before, alternating sequence of exons, etc. So genes vary a lot in size. In humans, if you see, average is 3000 BP, largest is 2.5 million BP. Genes are separated by sequences with unknown functions. Only one strand of DNA carries biological information, template strand. So potential to store biological information is enormous. So here if you see genes, this diagram illustrates. See, if you see here, genes are made up of promoter regions and altering regions of introns. In earlier, I have previously I have explained what are introns and the if you see what are introns, I will show you. Mm, Here, if you see, I told you all introns and the exons. Okay, so if you see here, let us go to the, yes, this slide. If you see here, exon protein coding sequence, intron is intervening sequence. Now, coming to this, if you see here, genes are made up of promoter regions and alternating regions of introns, non-coding sequences. Exons is the coding sequence, is the protein sequences. So the production of a functional protein involves in the transcription of the gene from DNA into RNA, the removal of introns and splicing together of exons, the translation of the spliced RNA sequence in a chain of amino acids. So these both plays a major role in splicing and the translation. Okay. Next, splicing. What is splicing? Exons are sequences of DNA that are expressed protein. Okay. Introns are intervening sequences that are not translated into proteins. Here, this is a diagram which illustrates that. So, if you see here in prokaryotic cells, bacteria, newly synthesized mRNA molecule consists of continuous sequence of codon that can be used by ribosomes to synthesize proteins. That is why the bacteria will synthesize vigorously. That is, prokaryotic mRNA is its fully functional and mature form and does not need to be modified following transcription. Genome and genomics, if you see, uh, now we are going to see about that. Previous one we have seen about genes and its different variations. So genome, what is a genome? The genome is all the DNA in a cell, which includes genes, intergenic sequences, and different kinds of repetitions. Specifically, it is a complete genetic constitution of an organism encoded in nucleic acids that is called as genome. So, eukaryotes can have 2 to 3 genomes: nuclear genome, mitochondrial genome, plastid genome. If not specified, genome usually refers to the nuclear genome. So, genomics is a study of genomes, including large chromosomal segments containing many genes. Nothing, genomics is nothing but it is a study of genomes, which includes a large chromosomal segments containing many genes. So, the initial phase of genomics aims to map the sequence and initial set of entire genome. So, functional genomics aims to deduce information about the function of DNA sequences. So, if you see, this is a small human genome project, its goals and milestones. If you see, identification of approximately 30,000 genes in human DNA has been done. Store this information in databases, improve tools for data analysis, etc. Milestones: 1990 project initiated as effort of US Department of Energy and the National Institutes of Health. April 2003, if you see, HCB sequencing is completed and project is declared finished 2 years ahead of schedule. So, if you see here, proteomics, what is proteomics? We'll see proteomes. Proteomes are complete set of proteins in cell, tissue, organism, etc. Okay, much of the information about proteins is not in the genome, that is the phenotype. So this illustrate like genomics, DNA tells what possibly, transcriptomics, RNA what probably, proteomics, proteins what actually happens. This is a thing. This is a flow chart. So if you see the genome versus proteome, if you see here, genome and the proteome, the image illustrates here genome 26,000 to 31,000, protein encoding genes, human proteins, if you see, less than 1 million. So this is a proteome complexity which this image illustrates. You can see the graph here. So if you see here genomics, the omics nomenclature, if you see the genomics and proteomics, what we are talking, this is called as the omics nomenclature. Genomics, DNA, gene, transcription, transcriptomics, that is RNA, which includes proteomics, that is protein translation process, metabolomics, that is metabolite. So transcriptomics and proteomics are the functional genomics. Genomics is DNA, that is the transcription. So a few definitions, if you see, gene, transcript, protein, metabol. So sequence of complete set of genes, transcripts, proteins, metabolites. Gene prot, that is analysis of genome and proteome. So proteomes, if you see here, proteomes, proteomics is a study of proteome. So the proteome allows for characteristics of proteins, understanding protein interactions, identification of disease biomarkers. So the identification, characterization, and qualification of all proteins involved in a particular pathway in a cell, tissue, organ, or organism can be studied by proteomics. So advantages of proteomics, if you see, the advantages of proteomics. So, unlike related fields like genomics, proteomics allows for study of post-translation modifications and interactions. So, why study proteomics? So, what is the need for it? It helps in understanding the structure and function of different proteins as well as protein-protein interactions of an organism, and it also is very important in drug development and understanding biological process, as proteins are the most favorable targets for various drugs. So this is a chart which shows the functional, structural, and differential terms. This is a proteomics flow chart. So proteomics has biomarkers. Biomarkers are biological indicators of a disease. They are useful both for diagnosis and response to therapy. Two major types of biomarkers of exposure and biomarkers of diseases can be done with proteomics. Existing biomarkers, if you see here, the cardiac region, the everywhere, the respiratory region, everything, this diagram illustrates this. Applications of proteomics. Mining, that means, as I told you all, identification of proteins. Protein expression profile, that means identification of proteins in a particular state of a particular organism. Next is protein network mapping, protein interactions and living systems, how it can be interacted. Mapping of protein modifications, and after interacting, how it can be modified. The mapping one, how and where the proteins are modified. So these are the applications where the proteomics is done. That is nutrition research in diabetes research, in cardiovascular research, autoantibody profiling for autoimmune diseases, urological cancer research, fetal and maternal medicine, neurology, renal disease diagnosis, study of tumor metastasis, protein biomarker. So protein classes for analysis can be done in membrane, soluble proteins, nuclear, chromosome associated, phosphorylated, glycosylated complexes. This is a flow chart which indicates the different analysis. So, current proteomics technologies, proteomic profiling separation, that is the separation stage, which indicates the two-dimensional sodium SDS page. And if you see, the liquid chromatography is a part of this profiling or separation. Protein identification here, peptide mass fingerprint, tandem mass spectrometry. It is termed as MS. Quantitative proteomics, isotope coded affinity tag, ICAT. So this is a chart which shows you this diagram. So thank you. This is about genomics and proteomics.